Hormones are the secretory products of ductless, that is endocrine glands, released directly into the circulation in small amounts in response to a specific stimulus, and on delivery in the circulation produce a response on a target cells. Classification of hormones depending mainly on their chemical nature. Hormones can be divided into peptide hormones, steroid hormones, and thyroid hormones. Peptide hormones, for example, anterior and posterior pituitary hormones, insulin, glucagon. Sex steroids are corticosteroids. Thyroid hormones and biogenic amines are, for example, thyroid hormones, adrenaline, histamine, bradykinin, serotonin, etc. Depending on the mechanism of their action, they can be divided into group 1 hormone, group 2 hormone. Group 1 hormones, these act by binding to intracellular receptors and mediate their actions via formation of a hormone receptor complex. These include steroid and thyroid hormones. These hormones have intracellular receptors, whereas group 2 hormones, these involve in second messengers to mediate their effect. These hormones have cell membrane receptors. Mechanism of hormone action. On the target cell, the hormone in combination with the receptor acts by any one of the following mechanisms. First, by altering the permeability of the cell membrane, or by activating intracellular enzyme, or through effect on gene expression, or through tyrosine kinase activation. Action through change in the membrane permeability. Hormone bind with the receptor present in the cell membrane, that is called external receptors, leading to its conformational changes causing either closing or opening of the ion channels such as sodium potassium channel. For example, adrenaline, noradrenaline act by this mechanism. Action through secondary messengers which activate the intracellular enzyme. The peptides and biogenic amines are two principal classes of hormones which act through second messengers. The hormones which act on the target cell is called as first messenger. This hormone in combination with the receptor form hormone receptor complex. This in turn activates the enzymes of the cell and causes the formation of another substance called second messenger. For example, cyclic AMP, IP3, or diacylglycerol. And the release of second messenger is mediated by GTP binding proteins, or they're also called as G proteins. So this is how secondary messenger is mediated by the hormone activity. Hormone called as the first messenger binds to the receptor. It activates the G protein, which is going to activate the effector molecule, which is going to increase the second messengers like cyclic AMP, cyclic GMP, calcium, and other substances, which brings about physiological response. Cyclic AMP as a second messenger. G protein stimulates enzyme adenyl cyclase. This increases the formation of intracellular cyclic AMP from ATP. Cyclic AMP stimulates protein kinase A, and protein kinase A phosphorylates enzyme protein. This enzyme is stimulated or inhibited. For examples, epinephrine, norepinephrine, ACTH, LH, GnRH, HCG, TSH are example through by which they mediate the action through the cyclic AMP. So here you can see the diagram of G protein, which separates from gamma and beta units, and upon separation or activation they convert ATP into cyclic AMP, which converts protein kinase into active protein kinases that brings about the cellular response. Inositol phosphate 3 and diacylglycerol as a second messengers. Here activated G protein, which is present in the cell membrane called GP protein, stimulates the phospholipase C. Phospholipase C hydrolyzes the phosphatidyl inositol diphosphate, that is PIP2, forming inositol triphosphate and diacylglycerol. IP3 and DAG are the second messengers here, and they bring out the response via protein kinase C. For example, For example, angiotensin 2 and vasopressin acts through these mechanisms. Here you can see peptide hormone, when it binds to receptor, they activate G protein, they activate phospholipase C, which converts PIP2 into PIP3 and DAG, which are going to bring uh, cellular response by converting inactive protein C into active protein C. Calcium colmodulin as a second messenger system. Many hormones act by increasing the calcium ion, which acts as a second messenger along with another protein called as colmodulin. The system operates in response to entry of calcium into cells, and calcium entry is initiated by changes in the membrane potential that opens calcium channels and hormone interacting with membrane receptors that open calcium channels. On entering a cell, calcium ions bind with the protein colmodulin, and colmodulin has 4 calcium sites, and when 3 or 4 of these sites have bound with calcium, the colmodulin changes shape, and this initiates multiple effects inside the cell, including activation or inhibition of protein kinases. Activation of colmodulin dependent protein kinases causes activation or inhibition of proteins involved in the cell's response via phosphorylation. Through effects on the gene expression, the mechanism of action of thyroid hormone is by acting on the genes on the target cells. Okay. So the hormone is going to bind, and they are going to bind to the intracellular receptors, and so this hormone and receptor complex is going to act on the DNA, and that is how it is going to bring about the cellular response. This is how the lipophilic steroid hormones also act, and the sequence of events here are the hormones are transported, like after secretion the hormone is carried to the target tissue on serum binding protein. Then there will be internalization, that is being lipophilic hormone easily diffused through the plasma membrane. So then it forms the receptor hormone complex, and it binds to the specific receptors inside the cell. Then there will be conformational changes occurs in receptor protein, leading to activation of the receptor. Then activated receptor hormone complex diffuse into the nucleus, binds to a specific region of DNA. This initiates transcription. Then binding of receptor hormone complex to DNA alters the rate of transcription of mRNA, and mRNA moves out of the nucleus and reaches the ribosomes. Here it promotes translation process. In this way, new protein are formed which result in specific responses. Then actions through tyrosine kinase activation, that is enzyme linked hormone receptors. These receptors, when activated, functions directly as enzymes or closely associated with the enzymes that they activate. These enzymes linked receptors have hormone binding sites on the outside the cell membrane, and their catabolic or enzyme binding site on the inside. When the hormone binds to extracellular portion of the receptor, an enzyme immediately inside the cell is activated, or occasionally it is inactivated. This mechanism of signal generation from plasma membrane receptors does not require G protein as an intermediate. For example, leptin receptors. The activation of tyrosine kinase can be activated by 2 mechanisms. One is due to the binding of hormone to the receptor. The receptor itself becomes a tyrosine kinase that phosphorylates the tyrosine residue on the intracellular protein substrate, and this sets into motion of cascade of events leading to enzyme activation and gene expression. Second one is hormone binding to extracellular portion of the receptor can attract intracytoplasmic tyrosine kinases like Janus kinase, or also called as JAK, and then activates them. This causes phosphorylation of transcription factors, proteins, and ultimately modulate the gene expression. For example, insulin insulin-like growth factors, epidermal growth factors, PGDF, ANF, GABA acts through